Hi children, in this video 4, we will be dealing with the factory or the host E. coli in detail. The widely used host is E. coli, or the popularly used host in biotechnology or RDNA is E. coli. And now we are going to see how the E. coli is going to take up the RDNA. So, this is the representation of E. coli, which is the preferred host in biotechnological experiment. Now, we have reached a stage in which the RDNA is formed, and as I told you, RDNA is the host plasmid DNA along with the gene of interest. So, host DNA plus gene of interest is called our rDNA. Now, it is a stage where this rDNA should get into the host E. coli. It is not easy for this rDNA to get into the host. The reason is that the DNA is hydrophilic. So, why doesn't it get easily into the E. coli bacterium? The reason is this: the DNA is hydrophilic. So, what has to be done is we have to make the host competent. We are making the host E. coli competent. There are different techniques of making it competent. The technique in the text is competence is by chemical transformation. Okay, so that is the technique we will be following, but when you go for higher studies, it can be also done by electric way, but we are following, or what is given in the text is the chemical transformation. So, let us see how this rDNA is going to be taken up by the host E. coli, and for that the host has to undergo a process called competence. We have to make the host competent to take up the rDNA. So, first of all, so this is the recombinant DNA. It cannot easily get in. As I told you, it cannot easily get into the host E. coli. The host E. coli should be made competent. This is going to undergo competence. Okay, so first of all, the E. coli is treated with specific quantity of some divalent cations like calcium. So, quantity is not mentioned, a specific quantity of divalent cations like calcium. So, the E. coli alone is treated with this, okay, with calcium. So, the first step is treating the E. coli with specific quantity of calcium. The second step is both the rDNA and the E. coli are made to incubate. First, they are incubated on ice. Immediately, they are subjected to heat shocks of 42 degrees Celsius and again back to ice. So, the second step is a sequence of incubation. So, the second step as a whole we can say is incubation. First, it is placed on ice, then subjected to heat shocks, and the temperature is 42 degrees Celsius, and again placed back to ice. Once it is incubated, both should be put together, the rDNA plus the along with the bacterium E. coli are subjected to ice, then heat shock and back to ice. This is incubation. This process is called incubation, and once that happens, pores are formed. See, you can see pores. Pores are formed on the bacterial wall, and immediately the rDNA gets in. Okay, so I'll make the rDNA get in. So we have the rDNA incorporated. This is how the rDNA gets inside. And the final picture, we have the rDNA inside the host. Now this is a transformed E. coli or bacterium or transformed host, and this process is called transformation, just a two-step procedure. Reiterating the steps, in the first step, what is done? In the first step, the E. coli is treated with calcium, specific quantity. Then second step, what is done? E. coli and the rDNA, both E. coli and rDNA are incubated on ice, use the word incubated. Then heat shocks of 42 degrees Celsius and then back to ice. What happens? Pores are formed and then our rDNA gets in, it's incorporated inside the host. And finally, we get the transformed E. coli with the rDNA. Clear? So, actually saying it's just two steps. First, treating with specific concentration of divalent cations like calcium. Then the next step is E. coli and the rDNA are incubated on ice, heat shocks 42 degrees Celsius and back to ice. By the time pores immediately are formed on the walls of the E. coli, the rDNA gets in and the pores are sealed off. And finally, we have the transformed E. coli. This process is called transformation, and this is done with the E. coli because here we use E. coli as a host. Sometimes we can use other organisms as the host. It can be a plant cell or animal cell. In that case, we can't go for this procedure of transformation. The transformation is different. So, other methods of transformation when we make a change in the host. If the host is E. coli, transformation is done by this. First, we are making E. coli competent. And I told the reason why, because DNA is hydrophilic, it cannot easily enter the host. See, does not easily enter the host because DNA is hydrophilic. That's why there's a two-step procedure to make the host competent. First, the host is treated with specific concentration of divalent cations like calcium. Then the next procedure is incubation, 3-step, on ice, heat shock and back to ice. Now what happened? The rDNA gets into the host and finally we get a transformed host. So this is the usual procedure with the usual host E. coli. Now I'm making a change, the host change. If we are using the plant cell as the host, if the plant cell is used as the host, the technique changes. In that case, we make use of gene guns. What is done is high velocity microparticles of gold or tungsten coated with the DNA, gene of interest, is made to bombard. See, it's made to bombard the target cell, which is a plant. So the procedure is like this. With the help of gene guns, what are the pellets? They are microparticles of gold or tungsten, and they are high velocity microparticles. They go at a very high velocity. They travel at a high velocity. So these particles made up of gold and tungsten are coated with the DNA or gene of interest, and they are made to bombard the nucleus or made to bombard the cell of the plants or the plant cell. So this procedure is known as biolistics or the gene gun procedure, and that is done in plant cell when the plant cell becomes the host. So usually it's E. coli, but now we make a change if plant cell is made as the host. On the other hand, if animal cell is made as a host, the technique is micro-injection. In micro-injection, what is done is the foreign DNA or the alien DNA or the gene of interest is directly injected into the nucleus of the animal cell. So that is micro-injection, where the host is the animal cell. There we don't make use of gene gun. In that case, we make use of micro-injection, where the foreign gene or the gene of interest is directly injected into the nucleus of the animal cell. So is it clear? And the actual picture of microinjection is this. I wish to zoom it. It's not like the normal syringe. Definitely there's an injection here. See, this is the microinjection, but it is seen under the high power of an electron microscope, and then correctly it's focused and the needle is made to touch, or it is made to be injected into the cell, and that too into the nucleus, goes down till the nucleus. So, there are the accessory structures to facilitate the correct target, that is the nucleus. So, this is the actual structure. Same way, gene gun, this is just a cartoon. This is the actual gene gun where it is correctly focused at the plant cell, and also you can see this eyepiece with magnification, and then only the gun pellets are shot or bombarded. Clear? So, this is in plant cell we make use of gene gun, and in animal cells we make use of micro injection. Yet there is another method of transferring the gene of interest, not with the help of any machinery. It can be done by another method called by the use of disarmed pathogens. In this technique, which is applicable to both plants and animals. So, now in this page which is opened up for plants, I am showing how we are going to disarm a pathogen, and deliberately we are infecting the host plant. Okay. So, we know the bacterium Agrobacterium tumefaciens. It causes tumors in dicots mainly. So, what we are doing, we are manipulating this Agrobacterium tumefaciens. Where it is manipulated? This Agrobacterium tumefaciens, when it invades the host plant, it produces a tumor inducing plasmid. And this tumor inducing plasmid, what happens when it is injected into host? It results in tumors in the plant. So, there are tumors. Have you seen some leaves with raised boil like structures like tumors? You can see mostly in dicot plants there are these tumor like structures. This is induced by Agrobacterium tumefaciens. When Agrobacterium tumefaciens invade the dicot plants, it produces, or it injects or introduces a plasmid called TI plasmid. TI plasmid, the full form is tumor inducing plasmid, and this plasmid induces tumors. The normal cells, normal cells undergoes uncontrolled growth and they form a tumor. And not only that tumors are synthesized, this plant slowly develops other symptoms like chlorosis, yellowing, wilting of leaves, then drying, etc., and finally it dies. So, with other symptoms. Tumors with a number of symptoms like chlorosis, drying, wilting, etc. Okay. So, now what the genetic engineers have done is they modified the TI plasmid. So, if I am drawing the TI plasmid in blue color, Agrobacterium is a bacterium, so I am depicting in the same way an oval with the chromosomal DNA and its plasmid. This is the plasmid, and the name of this plasmid, tumor inducing plasmid or TI plasmid. Now, the genetic engineers, what do? They engineered this plasmid, or they modified this plasmid. They identified the different genes. They identified the gene that brings about tumor. So, let the yellow be the gene that induces tumor. Then another part, I will give it in red, this gene that brings about all the other symptoms of pathology, other symptoms. Then some more genes, that brings about destruction to the plant. Now what did the genetic engineers do is they manipulated this. How did they manipulate it? Is that it was disarmed. So what we do is, I am disarming this which causes symptoms, I am disarming this. So finally they disarmed it, that is the symptoms, they made it non-pathogenic. They made it non-pathogenic, that's why I pasted off the genes that brings about the symptoms like chlorosis, wilting, drying, etc. And at the same time they did not alter the gene that induces tumor, that was intact, but on that gene they placed the gene of interest. Let this be the gene of interest. Here may be the gene of interest, may be the gene that produces the clotting factor. We know clotting factor is very important to be administered to those people having hemophilia, those who cannot, where the blood does not clot. We need the clotting factor. So we are going to produce clotting factor. So this is the gene that controls clotting. Okay. So this is our gene of interest, what I have colored in magenta. Now what did they do? They put the gene of interest on this tumor inducing gene. So the tumor inducing gene is yellow in color. So what they delivered the gene of interest on the tumor inducing gene, and now this bacterium is a transformed bacterium. Many manipulations are brought about. What are the manipulations? One is the gene of interest. Gene of interest is delivered. Next is they have been made non-pathogenic. So these are the two modifications. It is made non-pathogenic or it is disarmed. We can say it is disarmed. And at the same time the gene of interest is delivered. So these are the manipulations. And now this particular bacterium is deliberately made to infect the host. So this is Agrobacterium tumefaciens with all the modifications. Now we have the rDNA in it. Now this is made to deliberately invade the host. Made to deliberately invade another host. So I'll draw another host, a healthy plant. So by deliberately invading the host, it's invading. Now what happens as a result? It invades all the parts. Now what happens is this. Tumors are formed. See, I indicated yellow as the gene that controls tumor. Tumors are formed in every part of the plant. A lot of tumors are being formed because the tumor inducing gene is unaltered. And when the tumor inducing gene is unaltered, what happened? Our gene of interest, which I have indicated in pink, also multiplies in it. We know tumors are uncontrolled growth of cells. So this uncontrolled growth results, and at the same time what happened? Our gene of interest, which was pasted on this, will also multiply. At the same time, because it's disarmed, this plant will stay healthy with a lot of tumors, and the tumors having the gene of interest to man. Okay, so remember a list of D's. First D is delivering the gene of interest. Next D is disarming. The TI plasmid. And third D is deliberately made to infect the host. And finally, the desirable gene, which is our desirable gene, the color which I have indicated, and I am writing, desirable gene is multiplied where? In the tumors. In tumors. Clear? So disarmed pathogen goes by this principle, where the pathogen which causes disease in plants is the Agrobacterium tumefaciens. And how it causes disease? Once it invades the host, it introduces into the host a plasmid called the TI plasmid. And once this TI plasmid gets into the host, what happens is the host develops a lot of tumors, and not only along with the tumors they develop other symptoms like chlorosis, wilting, drying, all this types of symptoms, and finally the plant dies. Okay. Now what the genetic engineers have done, they modified the TI plasmid. How did they modify the TI plasmid is, they made it non-pathogenic. They disarmed the TI plasmid 1, and secondly they delivered the gene of interest. So two things they did. So once again I am reiterating the plasmid which was in blue in color, and there was already the tumor inducing which is yellow in color. This is tumor inducing gene. There are there's the other gene which produces the pathogenic conditions like symptoms. What did they do is they disarmed it. So I am plastering this, they disarmed it. So I am covering it with blue. So that got suppressed. Okay, this gene, the red gene got suppressed, its expression got suppressed. So the pathogenicity is suppressed, but the tumor will be induced because yellow I didn't ligate it or I didn't suppress it. At the same time, what did I do is the gene of interest. Our gene of interest is this gene of interest. And for example, this gene, let it be the gene which produces the clotting factor. Okay, this gene of interest is ligated on this. Okay, now what we get? Now what I got is an rDNA. In what context it's an rDNA? We got the gene of interest ligated. So when the gene of interest with the host DNA is called rDNA, and not only that, that is maintained. The tumor inducing gene is maintained. So the advantage is that in the tumors our gene of interest also gets multiplied. And lastly, what I am doing, I am incorporating it back into its host. We did the modification in the modification inside the plasmid. Now this host which is having this rDNA, now this host is called a transformed host or transformed bacterium or transformed agrobacterium. It's a transformed agrobacterium. This is made to deliberately invade the host. When it invades the host, what happens is plasmids will cause multiplication of the cells. That means tumors are formed, and when tumors are formed, what happens automatically? Our gene of interest is being multiplied. So we get this plant as a factory full of tumors, but the plant is healthy because other pathogenic conditions does not exist, like wilting and other symptoms which usually happen when an agrobacterium tumefaciens invade a plant. What and all are the other symptoms? That and all is suppressed, whereas tumors are formed, and inside these tumors our gene of interest also has been multiplied, and finally the gene of interest is extracted and the protein of interest is obtained. Okay, this is how the clotting factor administered for hemophiliacs are synthesized. Okay, so remember, other than gene gun, we can make use of disarmed pathogen. Similarly, in animals also the same procedure. Instead of micro injections, we can disarm pathogen. But here the pathogen cannot be agrobacterium tumefaciens. Here the pathogen is a retrovirus. You know what is a retrovirus? A virus which has RNA as a genetic material. So here what is done? The retrovirus is modified. How it is modified? It is modified in such a way to create tumors, but it is made non-pathogenic. So the modification is with two respects. The gene of interest is delivered. Then it is disarmed. So this is our retrovirus. And final D is, it is deliberately made to invade the host. And here the host is usually mice, to make a study of many diseases. Now this is made to deliberately invade the host, and finally the host mice develops a lot of tumors, but it is healthy. Why it's healthy? Because it's disarmed. And finally we get the gene of interest, which is incorporated into the retrovirus, also gets multiplied in these tumors. So remember the list of D's. As I told you, the first D is deliver the gene of interest. second D is disarming the pathogen, and last deliberately invading the host. So remember these D's, and finally our B, desired gene, is also multiplied where inside the tumors. So in a nutshell, other than E. coli, if the host is plant or animals, there are different techniques to incorporate the rDNA or the gene of interest. If it's plants, we can make use of, there are two options if plants are the host. One option is gene gun, and the second option is disarming the Agrobacterium tumefaciens. So two ways when plant is the host. Got it? So finally, when I recap it, the two ways when we make plant as a host, we can go for biolistics or gene gun, or we can go for disarming the bacterium, Agrobacterium tumefaciens. Similarly, if animals are the host, we can make use of two options. One option, as I mentioned, micro injection, and the second option is disarming the pathogen, and here the pathogen is not Agrobacterium tumefaciens, it is retrovirus. I hope you have identified. Under animal cell or animal cell, if it's used as a host, there are two ways of using animal cell as a host. Either you can go for micro injection or you can go for disarming the pathogen retrovirus. If plant cell is made as the host, there are two methods. The two methods are, one is the gene gun or biolistics. This is called biolistics. Or the other option is disarming the bacterium or pathogen, Agrobacterium tumefaciens. But the usual host is always E. coli. Widely used hosts are E. coli, and I told you rDNA does not easily enter because it's hydrophilic. It has to undergo a two-step procedure. One, it is treated with, the E. coli alone is treated with calcium ions, then later incubation on ice, heat shock and ice. So finally, in a nutshell, so other methods of transformation is, in a nutshell, to say if animals are used as the host, then the option is, one is micro injection, which involves an instrument. So this is an example for vectorless gene transfer. Then if plants are used as a cell, then it is biolistics or gene gun, which is also a vectorless gene transfer. On the other hand, vectored gene transfer, where it involves vector gene transfer, for animals it includes the second option, and for plants also the second option, that is disarmed pathogen. In animals, the disarmed pathogen is disarmed retrovirus, and in plants the disarmed pathogen is disarmed Agrobacterium tumefaciens. Okay, so there are two options coming under animals and plants. I have drawn it side or written it side by side. For animals, it is vectorless gene transfer is micro injection, and if it is vectorless, we have to go for disarmed. We have to vectored, we have to go for disarmed retrovirus. In plants, Vectorless gene transfer is biolistics, where we make use of an instrument, and when we have to go for vectored gene transfer, we have to go for disarmed pathogen, that is disarmed agrobacterium tumefaciens. So therefore the third technique stands like this way, common to both plants and animals, disarmed pathogen. But here you have to bifurcate for animals and for plants. This is for plants, it is specified, it is disarmed agrobacterium tumefaciens. Disarmed agrobacterium tumefaciens, and for animals it is disarmed retrovirus. So with this I think the topic is clear for you. So the usually used host is E. coli. Besides that, we can make use of animals or plants, and vectorless, it's micro injection in animals, and for plants it is biolistic. But we want to make use of plants and animals without using these instruments, then we have to go for disarmed pathogens. For plants, disarmed agrobacteriums, and for animals, it's disarmed retrovirus. So today's topic on host is over. I hope you have understood this video. Thank you.